Okay, welcome back. We are in chapter 4. We are discussing the anatomy of cells. We have just discussed the cell wall and portions of the prokaryotic cell that are exterior to the cell wall. And now we're going to discuss what is interior to the cell wall. So we start with the plasma membrane. The plasma membrane is made of a phospholipid bilayer. That means we have a layer of phospholipids here and a layer of phospholipids here. Phospholipids have heads that are hydrophilic. These phosphate heads are hydrophilic, and they have fatty acids that are hydrophobic. So they're able to make a barrier between water outside and inside because they don't cross that hydrophobic layer. But they're still able to interact with the water inside and outside through their hydrophilic heads. The plasma membrane, besides just having phospholipids, also has proteins in it. Proteins could be fully integrated into the membrane, which means integral proteins. These are proteins that fully go through the membrane. Or they could be proteins just on the periphery of the membrane. So it would be peripheral proteins. Proteins will help give structure. They can help affect the shape of the cell, and they can help allow things to go in and out of the cell. There can also be glycoproteins and glycolipids. Glycoprotein meaning sugars and proteins stuck together. Glycolipids meaning sugars and lipids attached together. Both of these can help protect the cell and lubricate the cell, keep the cell kind of fluid so those phospholipids don't pack together too tightly and freeze. Here's a micrograph. This is your cell peptidoglycan, your outer membrane, and then here is your cell membrane. So this cell with a thin layer of peptidoglycan and an outer membrane, what kind of cell would that be? Is that gram-positive or gram-negative? Hope that's something you can answer me. Make sure you're very comfortable with that. Okay. And then here's another picture of this lipid bilayer, an outside layer and an inside layer. Here are some proteins that are totally integrated into the membrane. They go all the way through. These are integral proteins. Here's a protein that's just at the side, just at the periphery. Here's a protein that's just at the side. It's a peripheral protein. Those are peripheral proteins. They can be involved in sending a signal on into the cell. They could be involved in helping do some sort of chemical reaction at the cell. This integral protein looks like it's a channel. It just makes a little pore to let things go in and out of the cell. So the cell membrane is often discussed in terms of a fluid mosaic model. So fluid means it's dynamic, it's moving, it's about the consistency of olive oil. And mosaic means it's made of different things. Those things it's made of are phospholipids and proteins. So a bilayer, a phospholipid bilayer with embedded proteins. Because it's fluid, the proteins are able to move around the membrane. Sometimes that's important to be able to function. It could open up to allow something to go through. It could shift to send a signal. Phospholipids are also able to move. They can rotate around. They can move laterally. Rarely you'll see it flip from one side of the bilayer to another, but usually it stays on that side. And the phospholipids movement allows for even more protein movement. The plasma membrane has some specific important functions. One is it's a barrier between what's inside and outside. It just physically isolates so that you can keep the cell's chemicals inside of it, and things that aren't the cell you can keep outside. It controls what comes in and out of the cell. So what membrane pores it has will determine what's able to go in and out. This is called selective permeability. Selective permeability is selecting what it's permeable to, selecting what can come in and out. So saying if you have an ion and a nutrient, you can bring it in. If you have a waste or a cellular product or some sort of signal you need to send, you can let it out. In prokaryotes, the plasma membrane is where nutrients are broken down or energy is produced. So without mitochondria and without chloroplasts, the plasma membrane is where a lot of that energy reactions are going to happen. And then the plasma membrane also offers support. It can help anchor cells to other cells and to tissues. So let's talk about some of the ways that things can go in and out of the cell membrane. If this is the first time you've talked about this, it might be a little complicated, so please feel free to come to me with questions or to spend some time studying, read your book, go watched some of those posted Amoeba Sisters videos or crash course videos. So the first kind of movement is simple diffusion. It's simple because it requires nothing. And it's diffusion because it doesn't take energy. It just goes right through the membrane from where it's a lot concentrated, really thick, to where it's less concentrated, it's less thick. So straight through the membrane. You don't need any sort of transporter. Goes from high concentration to low concentration. Does not use energy. Just happens all by itself. Simple diffusion is good for small molecules, especially nonpolar things that can interact in a hydrophobic environment. Oxygen, carbon dioxide, very small things. Things that are a little bit bigger or things that might have a charge are not going to be able to go straight through the membrane. So they're going to need a transporter. Some transporters are non-specific, just if it fits, it ships. If you fit in this channel, you can go right through it. Some are specific. You have to have this shape for it to open up and let you through. Facilitated diffusion is still going from high concentration to low concentration, still does not need energy. The only difference from simple diffusion to facilitated diffusion is facilitated diffusion now needs some sort of protein channel to get through. Osmosis is the movement of water. So water is going to go from high water concentration to a low water concentration. Although we don't often think of it like that, we think of it in terms of salt concentration. So water is going from a low salt or low solute concentration to a high solute concentration. So I think of it as water follows the salt. Water follows the sugar. Water follows the stuff. Whatever stuff is divided in water, that's where water is going to move to. So if you have this artificial cell, this artificial membrane, you have a bunch of sugars and molecules in here, and you don't have all the water molecules outside of the cell. The sugar can't move to come to equilibrium, so instead the water is going to move. The water is going to follow the sugar, so water comes into this cell, and that makes the cell start to fill out. So the cell bulges. Eventually there's so much water coming in that it starts to even move up. Eventually all the weight of this pressure, all the pressure and weight of this is going to start pushing water back out. So you're going to stop having water come in and you'll have instead water flowing in and out. That's reaching equilibrium. This pressure to push water back out is called osmotic pressure. Osmotic pressure is the pressure it takes to stop water from just coming in, but to actually push it back out. Water often flows through channels called aquaporins. So here would be an aquaporin. These just let water flow in or out of the cell at will. So if you have a cell in an isotonic solution, there's an equal concentration of solute inside and outside of the cell. So there's an equal movement of water inside and outside of the cell. You won't have a net movement of water. Your cell will stay pretty much the same size and shape. In a hypotonic solution, you have more solute inside of your cell than you have outside. Water follows the salt, so water moves into the cell. If your cell has a nice strong cell wall, water will come in and eventually that cell wall will provide enough osmotic pressure to stop too much from coming in. But if your cell wall is weak or damaged, as that water comes in, the cell wall doesn't have enough osmotic pressure to push the water back out. So water flows in and in and in and in and in and in until eventually that cell bursts. Then another kind of solution is a hypertonic solution. Hyper means high. So it's instead of having a hyper kid with a lot of energy, you have a hyper solution with a lot of solute. So in this case, water still follows the salt, but this time water moves in the cell to outside of the cell. So water is following the salt, and in this case, the salt is outside. So water flows out of the cell. That means your cell membrane is going to lose water and shrink down, pull away from your cell wall as it shrinks. This is called plasmolysis. And if this isn't remedied immediately, this is going to kill your bacterial cell. This is the basis of using sugars and salt to preserve food. Your food has lots of sugar and salt in it. So if any bacteria are left in that, can of peaches you just canned with a lot of sugar, or on that meat that you just covered in salt, any bacteria left there, the water in their cell will flow out into that sugary or salty environment. plasmolyze and die, and that's how your food is preserved. Sometimes your cell needs to move nutrients in or move waste out against the concentration gradient and is willing to spend energy to do so. This is called active transport. So active transport is moving molecules from where there's not a lot to where there is a lot. So it's moving towards a higher concentration against their gradient. This is always going to take a transport protein and always going to take ATP. And then group transport is a special form of transport that only prokaryotes do. So during group transport, they bring something in and then they stick a tag on it. And that tag says it is trapped in the cell. It cannot go out. And this allows your cell to accumulate like a nutrient, like glucose or something, accumulate it inside of the cell, even when there's not a lot outside of it. It's not going to flow back outside of the cell because it's now been tagged and is trapped. So you can tell me what agents can cause injury to the bacterial plasma membrane. You can tell me how are simple diffusion and facilitated diffusion similar and how are they different. You can describe osmosis to me. You can tell me what will happen to a bacterial cell if it's in an isotonic solution or a hypertonic solution or a hypotonic solution. You can tell me about active transport and group transport, what happens in those. Okay, I will see you back. Actually, let's finish this part out. Okay, let's talk about what's inside the cell. We have cytoplasm. Cytoplasm is mostly water, but also has other stuff, proteins, lipids, carbohydrates, ions. The nucleoid is where the chromosome is. The bacterial chromosome is frequently just one long, single, continuous circle, double-stranded, holds all of the genetic information. We've already discussed that. But in addition to its main chromosome, there may also be plasmids. There may be one plasmid, lots of plasmids, no plasmids. Plasmids are little. They're little pieces of DNA that are not part of that main chromosome, but they still have some important information on it to help the cell in special situations. There are ribosomes. Ribosomes make proteins, and they have 2 subunits on it. And you'll notice this is not addition, and that's okay, because these aren't telling you numbers. These are telling you kind of a combination of shape and size. It's where you would centrifuge each out. If you were centrifuging a cell, where you would find each cell piece. So you have a small subunit that's 30S, a large subunit that's 50S, and together it's 80S. Please make sure you pay attention to, a complete subunit that's 70S. Please make sure you pay attention to these numbers because they differentiate a eukaryotic cell from a prokaryotic cell. So a prokaryotic cell has 30S, small subunit, 50S, large subunit, and 70S altogether. These ribosomes are smaller and less dense than what you'll find in a eukaryotic cell. Bacteria also frequently have inclusions in them. Inclusions are like little pantries where they just have extra stores of something just in case they need it. One kind of inclusion would be a metachromatic granule where they store phosphate just in case they need extra phosphate to make ATP. Polysaccharide granules where they store glycogen and starch. They store polysaccharides in case they need energy. Lipid inclusions where they store lipid. Sulfur granules where they store sulfur. Carboxysomes where they store carbon dioxide. Gas vacuoles fill with gas, which allow bacteria to be buoyant in the water. And magnetosomes, you can see the magnetosomes here in this picture, are where they store iron, which makes them slightly magnetic. Bacteria can also form endospores, but only certain bacteria, Bacillus and Clostridium. These bacteria can form endospores. We mentioned these in our chapter 3 discussion of staining them. So endospores are a resting structure that some bacteria make. They make it as a way to survive when all of their nutrients are depleted, the environment isn't great, they're going to start dying. So instead of dying, they make endospores. And endospores are almost impossible to kill once they form. Endospores are resistant to desiccation. That means you can't dry them out. They're resistant to heat, so you can't just boil them out. They're resistant to a lot of chemicals, so you can't just throw an antibiotic at them. When an endospore forms, it's called sporulation. And when the endospore chips away and the bacteria starts growing again, when it goes back to a vegetative state, it's called germination. A bacteria can remain dormant in its endospores for up to thousands of years until something triggers that endospore coat to say, hey, it's safe to start growing again. So you could have Clostridium botulinum, or how about Clostridium tetanus? This is what causes tetanus. Clostridium tetani growing out on some old rusty nail in your yard, and it's just going to be there dormant until you come along and step on that nail, and that bacteria gets into your cell. Suddenly it realizes, hey, I'm not on this burning hot nail anymore. I'm in a place where it's warm and moist, and there's lots of good food here to eat, and it starts growing, and you've got tetanus in your cell. Here's how your bacteria sporulates, how it makes an endospore. So your bacteria, the first thing it's going to do is copy its DNA. This is important to note. It's not dividing. You're starting with one cell, you're ending with one cell. So this is not reproduction. This is just survival. It copies its DNA and starts to make this own little special part that's going to become the spore. It surrounds its DNA with plasma membrane, and then it starts to form a spore septum. You've got two membranes around it. You're filling it in with peptidoglycan, and then you cover this entire thing with a spore coat. And that spore coat is resistant to lots and lots of stuff. At that point, your original chromosome breaks apart, your spore is released, and you have an endospore ready to sit there and be dormant and survive until it looks like it's a good place to start growing again. Okay, so now you can tell me, where is DNA located in a cell? What are the functions of inclusions? And what causes an endospore to form? Okay, I'll meet you back for our next lecture video where we will talk about eukaryotic cells. See you soon. Bye.